on His Majesty's service. Neuroeffector Junctions. Synapses that transmit nerve impulses from neurons to effector cells, that is, the neuroeffector junctions, will be described in this video. The neuromuscular junction is a specialized cholinergic synapse that transmits action potentials from motor neuron to extrafusal striated muscle fiber. The vertebrate neuromuscular junction is composed of the expanded end of axon, the synaptic bouton, the specialized region of postsynaptic muscle fiber, the end plate, and several terminal Schwann cells. A fibroblast-like cells, recently referred to as kranocytes, in Greek kranos means helmet, represent the fourth junctional cell population. They lie outside the synaptic basal lamina, cap the neuromuscular junction above the terminal Schwann cells, and extend their cytoplasmic processes over the entire end plate area. Whether kranocytes play any role in synapse formation is not known. However, after denervation, they quickly proliferate and spread over the neuromuscular junction area before Schwann cell sprouting. Thus, kranocytes may play a role in neuromuscular junction repair after nerve injury. Neuromuscular junctions undergo extensions and retractions during muscle contractions, and it is noteworthy that the described organization of neuromuscular junction is maintained despite the continual mechanical disruptions throughout life. The synaptic bouton contains numerous mitochondria and synaptic vesicles filled with acetylcholine. Voltage-gated calcium channels are localized on the presynaptic membrane, and acetylcholine receptors and acetylcholinesterase on the postsynaptic membrane. The postsynaptic membrane forms junctional folds, which divide the primary synaptic cleft into the secondary synaptic clefts. Such a configuration provides an extension of the surface area, allowing a huge density of acetylcholine receptors and acetylcholinesterase molecules in a small space. Basal laminae of terminal Schwann cells and muscle fiber merge and continue into the synaptic cleft. It is known that axons have terminal branches. Thus, one neuron may innervate many muscle fibers. A neuron with muscle fibers which it innervates is termed a motor unit. The quantity of innervated muscle fibers by a single neuron depends on the nature of the muscle. Muscle responsible for fine movements, for example muscles of the hand, have motor units containing several muscle fibers per neuron. In the larger muscles, such as the quadriceps femoris, the ratio of one neuron to hundreds of muscle fibers may be found. All individual motor neurons that innervate a single muscle are called a motor pool. When a nerve impulse reaches the neuromuscular junction, the most events are similar to those that occur in interneuronal synapses. The voltage-gated calcium channels open up, which leads to the influx of calcium ions from the extracellular space to the cytoplasm. Synaptic vesicles fuse with the plasma membrane and release acetylcholine into the synaptic cleft by exocytosis. Acetylcholine molecules diffuse across the synaptic cleft, bind to nicotinic acetylcholine receptors which are ionotropic. Activation of receptors results in the opening of coupled ionic channels. The inflow of sodium ions causes lowering of membrane potential in the end plate, that is, generation of excitatory postsynaptic potential, the so-called end plate potential, which leads to the opening of voltage-gated sodium channels in sarcolemma, and muscle action potential generation. In turn, the latter triggers well-known events which lead to muscle fiber contraction. Acetylcholine is quickly broken down by acetylcholine esterase that prevents the continued stimulation of acetylcholine receptors or their repetitive activation. The nature of muscle contraction is determined by the number of motor neuron endings and by the number of muscle fibers that are depolarized. Depolarization of muscle fiber at a single neuromuscular junction corresponds to the all or none phenomenon, but not all synaptic bouttons discharge at once, which allows a graded response to the contractile stimuli. It is noteworthy that innervation of muscle fibers is necessary not only for their contraction, but also for their structural integrity provided by trophic influences. If the nerve supply to a muscle is disrupted, the muscle fibers undergo atrophy. As it was noted in the video about Schwann cells, terminal Schwann cells participate in neuromuscular junctions development and their maintenance in adult life. Terminal Schwann cells possess metabotropic muscarinic acetylcholine receptors and metabotropic purinergic P2Y receptors that regulate the release of calcium ions from the endoplasmic reticulum. During the neural activity, ATP is co-released with acetylcholine. Acetylcholine binds not only to nicotinic acetylcholine receptors on the muscle fibers, but also to muscarinic acetylcholine receptors on terminal Schwann cells. ATP binds to P2Y receptors on terminal Schwann cells. It leads to activation of the IP3 receptors of the endoplasmic reticulum and increase in intracellular calcium ions. In turn, it gives terminal Schwann cells the possibility to talk back to the pre- and postsynaptic elements to regulate the efficiency of synaptic transmission and release of neurotransmitters. Terminal Schwann cells can influence synaptic bouttons and muscle fibers via the release of different biologically active substances, TGF-beta1, agrin, and matrix metalloproteinase. Now about other neuroeffector junctions. Unlike neuromuscular junctions, which are always excitatory, junctions between visceral efferent neurons and cardiac muscle, smooth muscles, and glands may be either excitatory or inhibitory. Neuroeffector junctions consist of the terminal parts of autonomic nerve fibers and postjunctional specializations on effector cells. The terminal parts of the axons display the characteristic pearl necklace morphology with repeated enlargements or varicosities filled with various neurotransmitters. The latter include biogenic amines, purines, peptides, amino acids, nitric oxide. Cell membranes of effector cells possess corresponding receptors which are mostly G-protein coupled metabotropic, but there are some ionotropic receptors as well. Unlike the neuromuscular junctions, the junctional space in neuroeffector junctions is open to the extravascular space. Vagus nerves provide the parasympathetic innervation of the atria with some sparse ventricular innervation. The preganglionic fibers branching from the right and left vagus nerves reach the heart. They enter the cardiac plexus by synapsing with postganglionic neurons in the ganglia within this plexus. The preganglionic sympathetic fibers arise from T1 to T4 segments and partly from the T5 segment of the spinal cord. They reach cervical and upper thoracic ganglia, synapsing with postganglionic neurons, whose axons reach the cardiac plexus, innervating cardiomyocytes in the atria and ventricles. The postganglionic parasympathetic and sympathetic neurons innervate not only cardiac muscle cells, but also the coronary vasculature. Autonomic nerve fibers run in the myocardial interstitium parallel to capillaries and display the necklace morphology as was noted earlier. It was shown by three-dimensional two-photon microscopy that very often one cardiomyocyte displays contact with several varicosities from the same neuronal process and at the same time establishes contacts with processes of different neurons. The varicosities release various neurotransmitters, acetylcholine in parasympathetic fibers, norepinephrine and sometimes ATP or neuropeptide Y in sympathetic fibers. The M2 muscarinic receptors are the main subtype of acetylcholine receptors. However, M3 muscarinic receptors also play a physiological role in the heart. Adrenergic receptors in the heart are of the beta-1 and beta-2 types. All the above-mentioned receptors are metabotropic. The innervation of smooth muscles and glands follows the same patterns as that of all neuroeffector junctions. But one feature should be noted in the innervation of smooth muscles. In single-unit smooth muscles, one or several cells in a bundle are innervated by one autonomic nerve fiber. An action potential is propagated through other cells due to the presence of gap junctions between them. Thus, every bundle acts as functional syncytium, where all cells contract in a coordinated fashion. In multi-unit smooth muscles, each cell is innervated by an autonomic nerve fiber. These muscles are composed of cells that rarely possess gap junctions and therefore are not electrically coupled. An action potential does not spread from one cell to another, with every cell contracting after stimulation on its own. Thank you for attention.